participant screen sharing. Can you just allow me to share my screen, please?
You can share your screen.
Start sharing the screen.
Okay, got it. Okay, can you see my screen now? Hello?
Yeah, we can see the screen.
See my screen now?
Yes, yes.
Okay, great. Before I start, I want a small request from you. I hope you all have your mobile phones, smartphones on you or the desktop computer. I want you to do this for me. Can you visit this mentimeter.com? The participants, whoever is there, mentimeter.com, no need to sign up. Just on the top of the screen, you will see enter code to OAT. Can you see this?
Yes.
And please enter this code. Please enter this code. And vote. The options are given below. And submit it, please. I hope it's easy. Can you do this for me? Am I okay?
Yes, sir. No problem.
We need response from delegates.
Response from the delegates.
One person has responded.
Yeah. I request all the delegates to use that particular code, that is 202109713, and just
Vote. You can sit anywhere, anywhere on this planet. You can still vote. Doesn't matter. I'll give you another 30 seconds to vote, or we'll proceed after that. Okay.
participants, please log into that particular code.
Okay, this this is the best way to have an interactive audience. You see, we are sitting in isolated places at the moment. We can only do Zoom for obvious reasons, and hopefully you will be able to do the next question. Okay, there's one participant. It says only observations. 2 participants, measurements. 2 all of the above. Okay, can I proceed? Okay, there's one recordings. Okay, this is getting a bit interesting now. Okay, 3 participants, all of the above. 1, 2. Okay, good. Thank you for your effort in doing that. So I see that there is still not a good consensus on understanding what is data. Okay, to come back to the nuts and bolts of data integrity by itself. Data is facts and statistics collected for reference or analysis. That's the key. So the integrity part is a dictionary meaning of it, is the quality of being honest. It's the quality of being honest or having strong moral principles. Generally, they say that he is known to be a person of integrity. So, if you combine this data and integrity, the nut and bolt, the part that we are looking at here is trustworthiness of your data, and that's the washer. Because to firm up, even you can tighten the nut and bolt, But then if there is a washer, which is trustworthiness, the nut and bolt will tighten it and secure you to be honest. The idea is many of us get outliers like this. As you can see, these outliers are equally important than what is known to be as good data. I'm not sure how good a data is, but sometimes or more often than not, we always see students bringing up data which is too good to be true. That's the problem we all have. So the degree to which your presentation of facts and information can be viewed as honest and trustworthy. So if these two things are secure, you are automatically an honest person. But in analytical laboratory, the data integrity part becomes very important in terms of consistency of results and how are we going to handle this data. That's the key. So what we need is the completeness, consistency, and accuracy of the data. So this accurate data should be attributable, legible, contemporaneous, recorded, original, and accurate, which is also known as ALCOA, which is common term in industry. These are known as ALCOA principles. There is another one which is an additional to this ALCOA, but you have to understand the concept of metadata. Until unless you generally understand what is data, you will not have an idea of what is metadata. In the poll that I gave, what you voted for is all of the above. All the above components of the data are important to have data integrity. And the metadata is nothing but the contextual information required to understand the main data. So if you have data, you will have something additional to that which makes the data more clear and you can interpret in the way it has to be interpreted in a scientific dissemination. That's the key. So it is often described as data about the data itself. But then we also need to know how this data was generated. So when the data is generated, we automatically have something called an audit trail. Audit trail is nothing but a chronology of events where who, what, when, and why was recorded, especially an experimental value. How was it recorded? So the metadata will become the contextual information. Now, once you get the data, the contextual information, you have to structure it. The structured information describes the way we have handled or performed the experiments. And we also have to manage the data in such a way that it is retrievable. Once it is retrievable, then starts the traceability of that particular data with your experiment. So eventually, after all this process, we need to secure the data. So for example, if you look at, we are generally used to looking at HPLC chromatograms. So if you look at an HPLC chromatogram, we have a time, date, user ID, instrument ID, which gives us an indication how this particular experiment was performed. So the ALCOA principles become very, very important and in this context. Now, if you go to the ALCOA principles, it clearly indicates who recorded the data, whether it is permanent, for example, after it was recorded, and what kind of hieroglyphics are used. For example, if you use alpha or a beta or some kind of a symbol, what are the explained processes, and then if properly corrected, what is the procedure for correction? And data must be recorded at the time it was generated. That is the critical aspect of any analytical output that we generate, and close to proximity to its occurrence. So if you have performed an experiment, for example, a TLC, thin layer chromatography, you do cut the plate and then you spot and you develop the plate. Now, what will you do with the plate? You have to actually make sure you are labeling it right. Once it is labeled, it has to be chronological order, it has to be dated, and then laminated onto your book, for example, or the laboratory notebook that you are using, or a storage platform where nowadays you can do digital storage. And these things have to be kept in a consistent manner and performed religiously. That's the important part. The original data must be preserved, which is in its unaltered state. That is very, very important. And then this original data should have certified copies. And then more often than not, obviously, without any doubt, we need to have accurate output of this particular data whenever we perform an experiment. So it should reflect the action, observation made, and data checked where necessary. If there are modifications, then we need to make sure that we have an explanation ready for that particular system. Now, in addition to what we practice as Alcoa, there is Alcoa Plus. These are the components which add on to what is already described here. Available, enduring, consistent, and complete. Without being complete, there is no point in submitting the data. So it's all about data. The question is, how well do you know your data? If you don't know your data, you can't disseminate the data in any forum for that matter. It is very important and critical that but the person who actually performs the experiments should be aware of how the data is processed. Until unless we have this data ready, the path to paper being accepted, which is the bread and butter for all academics, everybody wants to publish, and people are waiting to smash you if in case you don't follow any of these components. So that is the critical level. Now, if you look at data itself, the data, what you actually generate, lives in two different forms. One is static form, one is a dynamic form. So static is indicated to say that this is a fixed data record, such as paper record, for example, or an electronic image. That's a digital image, for example. However, when you look at the dynamic part of the data, it's the format that allows interaction between the user and the record content. For example, you reprocess a chromatogram, so it is not anymore static, it's a dynamic one. It actually keeps changing, the area changes, or probably the theoretical plates may change, or probably the alpha, the capacity factor of the column may change. So that is the dynamic part of the data. So in a chromatographic record, you can allow, let's say, you change the baseline, or you attenuate a peak. Then what happens, or you inhibit integration. Now that is dynamic data. So you need to understand the difference between a static data and the dynamic data. The biggest problem that we have in general is presumption and assumption. There is no room for such things in research. If you design your experiment, perform your experimental, you generate, generally, most of the students that I have come across, they say my data is good. Now, what is good? Now, I don't want to hear the word good, because good doesn't mean anything to me. It has to be accurate, number one. Number two, the data should be relevant to the experimental that you have conducted. Basic problem here is we are involving ourselves in p-value research. You can actually, this is a statistical way of looking at it, but absolute research, when it comes to fundamental research, you have no choice, but you'll be exposed if you actually make any kind of malpractice, or you deviate from the experimental value. So it is very important we have to follow integrity and inculcate integrity. But it is not a new concept per se. Data integrity has been there for a while, and it's been a long time since we have known data integrity. And most of the data integrity guidance documents that we come across or CFR 211 US FDA. CFR is nothing but Code of Federal Regulations. And I'm not sure if you want to know what is, I hope you know what is 211, right? It is about good manufacturing practices. As you can see, in those parts and the sub parts of 211, it requires that backup data and exact and complete and secure from alteration and inadvertent erasures and loss. So you can't have a situation where you by mistake you have done something. Now that is not accepted in any research forum. For not necessarily only industry. Industry, it is a military rule that you can't have this kind of situation. But in research labs, it is the same thing. If we can follow in our day-to-day research work, obviously, you will have a good name for yourself, most important. And I will not go through all these documents and all these subparts because these are available in plenty. And US FDA website itself, it is available. You can go through all these things for yourself. Okay. Now, in terms of the concept of data integrity guidance, the regulatory agencies actually it started with MHRA about the GMP of data integrity. They gave a definition and guidance for industry. And then in 2016, WHO released one guidance. And again in 2016, April, US FDA also gave, released a guidance. And eventually in August.
कम्प्लीटच केलं. हा व्हिडिओ पण व्हिडिओ म्हणजे स्क्रीन कास्टोमॅटिक वर
Sorry?
असे लाईव्ह नाही केलेले नाही.
Can I continue? Is somebody asking a question? Hello?
You can continue, sir. It was
okay. Sorry, I thought somebody was asking a question.
No, sir. No, sir.
And then PICS, you know what is PICS. So they have come up with another guidance. So these are up to 2018, we have had various guidances being updated and promulgated. So these are basically, you have to understand, these are guidances. Regulations are different from guidances. Act is different from a guidance. Regulation is different from an act. So you have to understand. So I will not go through those aspects because that will take away a lot of time. And the question and answers for the guidance to industry regarding data integrity is available, from this particular agency, that is CDER. People who are interested are welcome to go to, that kind of website and look for information. But the important events that come in a DI cycle is, of course, I explained it to you, which is metadata. Then you have static versus dynamic records. And then how are you going to store those records? That's the backup data. And then the system validation itself, whether the system was validated for its intended use. And then, of course, comes the audit trail. When does the audit trail comes? When you write a thesis, right? In terms of your academic profile. So you need to understand the concept and the sequence of events, which is very important. Now, I'm giving you 1 minute to go through these chromatograms, especially the parts that are typed here about the chromatogram, the sample information that is. I'm not actually interested, or probably you could go through this, but what I want you to look at is have a look at this and this, the chromatogram A and chromatogram B. I'll give you 30 seconds. And now I'm going to ask you a question on the Mentimeter again. Which chromatogram of the ones which I showed you is DI compliant? You can go back to the Mentimeter and use this code, please. I hope you have seen the chromatogram. you want to see it once again, the code is up there. This one, okay, I'll put it up here. Is this okay? You can go to Mentimeter. Here is the code. Tell me which A or B, which one is DI compliant, data integrity compliant. You can vote, please, and start seeing. B, 1% B, 1% A, 2% B. Okay, interesting. I didn't expect this. Honestly, I didn't expect this at all. I'm really surprised. Three, okay. I'm getting very interesting. Okay, I'll put the Mentimeter up. Three of them say compliant, 2 compliant on the B. Anyone else want to try? Because this is critical, you see. I'm giving you 1 more minute to try, please. If anyone, I don't know how many participants are there. It would be nice to get bigger sample value. Just to get statistical significance. P-value better, isn't it? Trying to get a better p-value. Okay, 3 and 3. Wow. Wow. This is extremely interesting. Anyone? Finished? Four. Okay. Okay, let me explain. So I have 4, as you can see, out of 7 participants, 4 have indicated A is DI compliant and 3 have indicated B is DI compliant. So I'm taking the screen away. As you can see, first and foremost, if you see the sample type unknown, that's fine. And you can see 12, 27, 50 seconds, whereas this doesn't have it. So obviously, A is more compliant. If you look at this date processed, Sunday, August, whatever, 2005, at this particular time. So timestamp is there. You understood how it is done. So obviously this B is not data compliant because we don't know at what time this was processed or what time this was acquired. It was only the date, the time is missing, as you can see. And then if you look at the result table, so all you need is the area and percentage area and height. Here you have so many blank spaces, which is obviously questionable. Why was it required if it is not? So the written procedure is wrong if you have blank spaces. If there are spaces, then you have to have something filled, or you have to make sure that as per so-and-so SOP, that is not supposed to be filled. So you got some kind of an idea now to say that what is compliant is dependent on the timestamp, the date, and the processing parameter. Okay. So just to give you an idea. So the problem areas generally with HPLC analysis is security. The access control. Say if one person has done a particular experiment or analytical procedure, for a experiment. Another person using the same equipment should not have access to that particular procedure, which was done by another analyst. So obviously we need to have login systems, password protection, unrecorded, undocumented activities. You can't have, inserting USB drives, flash drives, or hard drives, collecting data. So you need to have a certain kind of procedure in place where you make sure that no one alters your data. And the other thing that is going to happen is repetitive testing. So if a certain test failed, so you repeat it again. So you actually are not looking at why did it fail in the first instance. And then the root cause investigation. This is very serious in industry. We actually have audit trail and timestamps. There are so many filters, and that is the reason the industry has moved away from regular book entry, logbook entry. Everything is electronics. So it is known as laboratory management systems, laboratory information management systems, or LIMS, and then laboratory notebooks where everything is electronics. So you can't have modifying the digital clocks just because you want to reprocess on a particular day and time. You can't just change the computer's digital clock and say, okay, I reprocessed at that point in time. So these are not a good, dishonest ways of doing it. And then you can't store in different files and different file names. So you need to have a very systematic way of storing these documents. And then the data management system has to be very clear. That's the key. All laboratories should have data management system in place. Institutional policies have to be created for that, how the data management works. Because the reason I tell you, all the thesis that probably your students or students themselves who are writing is going on to a website called Shodhganga, right? I hope I'm right, because I never accessed that website, but I am aware of that. Now, you can get any, I believe so, you can get any particular information about, any thesis that is being submitted in India, in Shodhganga. So can you imagine if somebody has plagiarized or altered or, created a document which is, falsified, it becomes an embarrassment to the institution, the person. So we will have a situation where we need to understand the gravity of the implications that everybody will have, including the supervisor. So it is very important that you do a periodic inspection of the search results. Is my screen visible? Hello? Yes? Can somebody do a thumbs up?
Yes, sir.
Is my screen sharing okay?
Yes, sir. Yes, sir.
Okay. Brilliant. So in terms of every time that we have to implement the principles of ALCOA, and then data quality review is critical here. Either it is paper or audit trail, which are electronic, it doesn't matter. Consider, I'm telling for institutions, it is very important to understand, especially quality assurance. Quality risk management is very important. You can get a lot of information from ICH Q9. It's a very good practice. Please try to understand that and make a attempt to implement those systems. But in case of analytical methods, it's very important to have consistency. Regulatory expectations are not, very important for academic institutions, but UGC rules must be there, obviously, or AICTE rules or norms that are, we are bound with in terms of how the consistency and stability of the experimental data is produced. And then in terms of industry, FDA expectations is very, very, they're very serious about this. In terms of, is the reprocessing procedure available, analytical method should be validated. And you know that very well. You can't have 20 injections, say, okay, I finished an experiment. You need to have a validated method. And then for that, you need to have a table of contents. You need to have a spreadsheet. You have to make sure your data is secure. So the supervisory procedures for the review of reprocessed data, it becomes very, very important. The supervisor should be aware of handling the data so that the student performs an experiment and then collation of data and securing the data is done in a very proper and a methodical way. Okay, what is a research audit now? Now, a research audit is nothing but an audit trail. It is the importance that is given to chronological data. If you perform an experiment today, we need to have a timestamp reprocess. Say, for example, you conduct an experiment, HPLC experiment today, and then after 1 month you reprocess. Now that is questionable again. You can't reprocess after 1 month to understand or to actually use the data to calculate the output, whatever it is. It could be percentage assay, it could be a dissolution, output. You can't do that. You have to do it contemporaneous. You have to do one after the other. So the DI requirements are data generation, data transfer, and data deletion in case it is acceptable. So it is very important. So you need to have the audit trail in such a way that these are all autonomous operations done parallelly. So remember that. And then what it captures is overwriting, misrepresentations. You suddenly abort a sample run and then run another one. It is not of a particular sequence. Deleting some runs because it is not convenient for you to have that data. Like I told, data alterations, there are many FDA 483s that I can show you. And then repetitive testing till you get a result that you want, you keep on testing it to obtain compliance. Now, how to perform a research trial? You need to be having electronic record system. QA could, work on it. In industry, especially, FDA recommends to review each record. It is a normal procedure. The final approval of the record is done by quality assurance. And then, like I already mentioned and reiterate myself again, is the chronology of events, sample testing sequences, identification and tracking, critical process and analytical parameters. Every time it has to be followed as per the SOP. That is non-negotiable. Now, in the current scenario, we are possibly not storing much data in a paper form, in a written form, that is a printed form. Either you write it or you print it. We are not storing much data in such thing because we want to conserve, paper, and we don't want to have paperwork carrying files. Now we can transfer files. But then, if you are storing digital data, there is something called a computer system validation, which is a very key component of data integrity. These validation, these procedures, CSV procedures, the SOPs, the system nomenclature, the use and validation are all provided in the good automated practices in EU, that is European Union Annex 11 and FDA 21 CFR Part 11. You're welcome to visit these websites and you'll get more information about this. There is abundant information. It will take you a month to read all these things. And if anyone is really interested, then you can go to ISPE, where GAMP 5 is there. You can see what is computer system validation. That's probably the most accepted practice in industry. It's also known as GAMP 5. So you could consider that. Now, regulatory aspects of data integrity. We have seen what is data integrity, why it is important, but then the regulatory aspects. See, I did tell you in my previous slides that guidance is something that you can follow. That's not an issue. But then how you follow is up to you in any case. But regulations are non-negotiable. You have to follow them. That's the key aspect. Now, if you actually look at working on a HPLC data, system suitability is very important to make sure that the HPLC system is performing to its optimum for a given procedure. So now if you actually go and look at it, so the testing, sampling, FDA prohibits. See the wording, it's very important. The regulatory wording is very critical. You need to understand the communication of regulatory systems. FDA prohibits sampling and testing with the goal of achieving a specific result. That means you can't test things because you want particular results or to overcome an unacceptable results. So this practice is also referred to as testing into compliance, not consistent with CGMP, that is current good manufacturing practices, which is nothing but CFR 211. Now, we can go into a lot of detail, but then these are available for you to read. You will probably have better understanding. This is just to give you an idea of what the DI concepts are in industry. You can probably go through this and in much more detail in the respective websites. Say, for example, why do we perform system suitability for an HPLC experiment? I don't know how many of you actually perform an experiment by using HPLC, but this is an example that I have taken because generally almost all institutions have HPLCs nowadays, and most of the experiments and the data is generated from an HPLC instrument for that matter. So the chromatographic studies, it should be performed according to the firm's established written procedures. So if you're in your case, for example, in academics, it will be institutions established written procedures. That include the identity preparation to be injected and the rationale for selecting that particular method. That means it has to be validated. It's simple and straight. And then you need to have standards. If you don't have a reference substance, which is a qualified one, you need to characterize the secondary standard with written procedures, and the sample that you are characterizing should be from a different batch. You cannot have a standard and a sample from the same vial or a sample bag. So you need to understand the difference between a standard solution and a system suitability solution. These are something that it's very critical and you build into it. Now, many of you probably know what is a just a minute. So now that I have given you an indication of what is an audit trail, before we go into FDA 483, please take this voting and give me an answer, which is on your screen. The code is given here. Are you able to see the screen? I'll wait to see how many people have. All of the above, very good.
Yes, sir.
I'm sorry, I switched. There you go. Data generation. Interesting, hey? So that is why I asked this question after I explained what is an audit trail, so that I wanted to see how well it is understood, the concept. 3 of them have attempted, 2 have told all of above. 2, 3, okay. Count down. 4, yes, okay. 2 have done data generation, 2, 4, 6. Okay, the answer is all of above, actually. I did mention in my previous slide, right? So So we close this voting. So 6 all of above. That's very good. Okay, I'm asking you one more. So let me see, after having spoken for almost probably 20-25 minutes, let me see how you'll give answers for this one. Critical aspects for DI requirements are. The code is here. You can see this code. Data generation.
I request delegates to respond.
Yeah, I'm waiting. 2. Data generation. All of above. 2, 2. 3. This is real time, so you are seeing what I'm seeing, right? Your people are voting, and we can actually see how the voting is being done and how the answers are selected, options based on the options.
Delhi.
Are you understanding the concept here? Because in industry, this is survival. If you don't understand data integrity, you will never get a job. You will be thrown out, literally. It is the same question I asked. The previous question and this are same. There is no difference. I just twisted the question. The concept is same. It is all of the above.
Talk. Delegates, I request please respond to the speaker staff. It is a real-time data generation. Sir is giving a live example, which will help you to understand the concept clearly.
My previous question was same. Again, there was a discrepancy in answering. I'm surprised that you could answer data generation. Critical aspects are all of the above. So obviously, you need to understand. That is the reason it becomes important. Is you need to keep reading regulatory documents, because the way they write, the way it is expressed, the language is very important. If you don't understand the language, this English in regulatory systems, the English is different from what we generally talk. Please understand that. The regulatory is legal. It's a legal terminologies that are used. So if you want to understand, you need to comprehend the legal terminologies of regulatory systems. Okay, so I'm taking the thing away. Thank you very much for the voting. And I'll just give you an idea of how actually the warning letters are given. Say, for example, there is a warning letter. It's a 483. All 483s are not warning letters. Mostly there are observations. See, 483s will be like this. FDA 483s. So you will have inspections. So if you look at the Office of Regulatory Affairs, electronic reading room, you will find a lot of warning letters. You will have observations, inspections, everything, all are listed here. It's all there on the web. It is updated almost, as frequently as I can remember. And each file will have, for example, for year 2020 Excel file is also there, observation data sets. So all 483s are recorded and observed. So the warning letter, for example, it will look like this. For example, I'll show you an Indian context. This was in April 2016. It is there in the web. It's not something that I have made. This is a US FDA website, a warning letter by the US CDER. I can try and improve the resolution here. Can you see this? So the warning letter number will be there via a courier, registered courier. So they will give complete observations. Our investigator observed specific violations included, but not limited to the following. This is the, again, I am telling you, this is the legal way of putting it. The wording is very, very critical here. So we reviewed your firm's response data. They have already responded. See, always remember, when you get a 483, it is an observation. Within 15 days, you have to respond. When you respond, if they take it up, from there on, it becomes a warning letter. Okay? So it is very important how the meaning of adulterated drug can be applied here under section so-and-so of the FDC Act. That is Food, Drug and Cosmetic Act. Remember, I did mention about this. Act is different from regulation. Regulation is different from guidance. Act and guidance, regulations are three different systems altogether. So you need to understand the CGMP implications here. So what did they do? What did the analyst do? A QC analyst injected 11 identically similar named samples for impurity and assay analysis, approximately 1 to 15 seconds apart from one another, according to HPLC audit trial. You understood? The observation. After that, a second analyst injected 8 similarly named impurities approximately 12 to 16 seconds apart. Can you see why the time stamp is so important? I did show you a chromatogram to make you understand how the timing is put in a chromatogram and why it is important. So this is exactly what happens. And then what did they do? Neither analyst reported the results obtained during the testing. So both analysts have performed the experiment, but they didn't report it. But the digital data is there in the computer. So the inspector found it. You understood? So now the laboratory incident report. We always have an incident report that is done. By the first analyst, he or she has deleted 28 original files due to pressure fluctuations. It happens in HPLC, ghost peaks, or whatever could have happened. While the second analyst deleted the original injections working standard associated with peak shape. The problem was because of a peak shape. Now you see what happens to the company when you get a warning letter. So both analysts here have changed the clock prior to analyzing the samples. That means they were falsifying results. They were dishonest. It's a malpractice. So that is why data integrity is very, very important. Data integrity cannot be built into an experiment. If you have integrity, then your data is trustworthy. You understood? So now the QC analyst injected another sample, and then the injection file was saved, had been deleted afterwards, the original sample. The response acknowledges that the analyst deleted 8 injections, including all the samples. Now, this is how systematically they write observations. That is why US FDA inspectors are the most difficult audit trail inspectors that you can ever find. It's not easy. They will go to any length to find what is wrong. Once they find it, you're done. So you did not include the metadata with the audit trails in your data backup. I did tell you, you need to have data transfer and then backup, and then you have to have a system. So the metadata, which was part of the chromatogram, was not included in the audit trail. Now, the system crashed, the HPLC system crashed, and all data is lost on the instrument, including audit trail information. Now, even though the system backup was available, the response that they gave in the first observation, 483 observation, the backups are performed. There are SOPs, but it was not done. So this is how exactly they go through each and every aspect of how data integrity is put in practice. So the passwords, if you go to passwords, you can't have admin passwords. You can't have users. You can't have one login password for everybody. They have to be user specific and passwords. So you need to understand. So this particular thing is available on this web link. There are many. There are hundreds. You can go through all this. Each and everything, not necessarily HPLC, even Car Fisher auto titrator was not restricted, for example. So they'll go through every small detail, and once you are caught, your firm failed to follow written procedures and designed for processing quality control. At the same time, there is malpractice. And even worse, what happens generally is our investigator discovered that your firm was destroying original batch records, backdating revised implements. Can you see this? This is a normal thing, in smaller firms. They just tear up the records and throw it away. What I'm trying to tell you is not to impress upon what industry does. Even in your own research that you do as thesis or experimental or whatever you do, if you don't record it properly and be systematic in your way of keeping things, obviously you will get into trouble when you actually have a job. So this is how they do it. In conclusion, these are serious violations. So they will go through everything and then they will probably issue a legal notice also. So it could be anything. If there is a death, then it is finished. It is done. If somebody dies because of this situation, then we can write off the entire, effort, whatever the company and everything. So you need to understand the seriousness of these things. And then the person who actually collates is listed. Each file will be handled by different inspectors based on their assignments. So you need to understand how this works. And like I did mention, within 15 days of this receipt of this letter, please notify that you have taken correct actions. If not, they will blacklist or whatever you call it. They will put you on alert on the system. So this is exactly how it works. So please understand, for us in industry in general, data integrity is bread and butter. FDA warning will always be there. Peak inhibition and peak integrity and other things. And the deviation cited, you are responsible for investigating these deviations. And obviously, you can actually argue if you are good. And there are many instances where people who have been issued letters have argued their case and won also. That is, if you are good at it. So, in conclusion, introduce DI systems at college and university level. It's very important. This ensures quality of work and output, obviously. The most important thing that I would like to tell you is confidence. The confidence in the experimental performed. Even more important is the reproducibility and repeatability of results. What is reproducibility? What is repeatability is something that you need to understand. Repeatability is nothing but if you perform your own experiment, you will be getting consistent results. Precision and accuracy, obviously. Reproducibility of results is if your experimental data is performed by another person in his own lab or in a different scenario, they should be able to get the same results or similar results or close results, which are statistically acceptable. What is more important with this particular reproducibility and repeatability? Eventually, if you have data integrity, your reputation as a person, as a scientist, as a supervisor, or as an industry professional will be very high. And here is one particular slide I would like you to focus on. Is this is published in Nature. I have given the website here. Nature. It's a very beautiful. So generally, what factors contribute to the reproducible research? This is the key. Always, often contribute, sometimes. So the lighter version is sometimes, and always is the darker version. Selective reporting. That means you choose results which are good. I meant to say good. I don't know what it is, but if the supervisor, your guide accepts the results, then means it is good. That's where it goes to. Okay. And the pressure to publish. If you want to publish, you push any result. Low statistical power, obviously p-value research. There is no end for that. Literally everybody does p-value research, and that is something that I personally avoid, and cannot be replicated in the lab, in the original lab. Insufficient mentoring. Mentoring is very critical, and most of the universities abroad, they always have mentors to work with. And methods, the SOPs, code means SOPs are unavailable, poor experimental design. So obviously, when you look at this entire scenario, you're looking at the first two aspects is more than sufficient: selective reporting and pressure to publish. So don't put yourself in a situation where just because you want to somehow get the thesis out, or somehow you want to publish, or somehow you want to convince, don't do selective reporting. Data integrity is critical. Your own integrity is even more important. Your name is important. You can work for 20 years and create a good name for yourself. One paper which is plagiarized or which is falsified data can kill your career. And I know personally, some of my acquaintances have gone through this and they have lost their jobs. So universities abroad are very, very serious about it. One shot, they will just clear it up. So please be aware of it. And thank you very much for the time and your attention. If there are questions, I'll be happy to take it.
Delegates, please ask the question.
Where do I see question and answer? Is there
It will be in the chat box.
segment or somebody just asked me, how is it done?
Please ask the question.
See, industry is a completely different ballgame. There the systems are different. They are more streamlined and implemented in a very religious manner. However, in academic institutions, there is no single rule, you see. And what happens generally is it all depends on the supervisor. That becomes a bit of an issue. So I wouldn't be able to comment whether you should follow or not follow, but in general, everything has become electronic nowadays, and people have access to so many, publications, and we can't escape. There was a time when you can publish in some, remote journal and say, okay, I have published, but now even that is accessible. So make sure you're aware of things. And obviously, if you have a good record for yourself, people invite you to uh, write papers or to collaborate and generate the kind of uh, research work that you do. And I'm sure your principal at KLE, College of Pharmacy, is a good friend of mine, and he has seen how we work and how things are put in place and how the records are maintained. And probably he's a very good person for you all to interact and understand how to take it forward in case of in advisory capacity. He'll probably give you more hands-on experience that he has had at Rhodes University on his research.
There are no questions.
Yes, tell me. I'm fine. I can't see you, but I can hear you very low. Can you speak out a little bit loud, please?
Can you hear me now? Sir, can you hear me?
Yes.
I am Manur here, sir.
Yes, Manur.
Sir, really, the session is very much informative for all the delegates. There is a topic titled "The Importance of Data Integrity and in Analytical Laboratory."
Thank you.
The presentation of the topic is exclusive to with all the relevant examples. Very which is unable for the delegation, sorry, everyone to access for such type of examples, which usually they don't look into it. And you have made an effort to collect all the relevant examples, which is very much important. As you said, the data integrity is bread and butter for the research. So with all the nice examples you have given, I hope the delegates have understood clearly what sir has mentioned about the data integrity and its importance when we are doing any research, especially. Thank you, sir. Thank you for accepting our invitation. And I request V.S. Master sir to conclude the sessions.
Good afternoon, everyone. So thank you, Dr. Srinivas, sir, for enlightening us on the data integrity. As rightly said by Dr. Srinivas, data integrity ensures the recoverability, searchability, traceability, and connectivity. And also, along with that, it also helps in improving the performance of the industries, as well as it also helps in taking the right decisions based on the data and all those things. With this, I come to the end of this session. Thank you, Dr. Srinivas Patnala. Hope you guide us or you come in the long days. We will be seeing you for the further sessions. Hope you keep in touch with us.
It's always a pleasure and good to see you all also. It's been a while. I hope you're all doing well. But at the moment, I teach, I'm part of the education program at the United States Pharmacopoeia. I'm a consultant. So I train fresh graduates and industry, people on quality control methodologies. So we in fact teach in a very systematic way. And because of COVID, we have stopped sessions at the moment. And it's a very intense program. And I'm sure, as we go by, you will probably see more such programs and work around it. And give my regards to all our colleagues there. and
dear participants, if you want to interact with Dr. Patnala, we will be providing his email ID. So
Yes.
the question you can directly speak to him, and he's always available to you, and he can answer your queries. Okay?
Thank you.
Thank you. Thank you, sir. Thank you.
Thank you. All the best. Have a good time.
On behalf of KLE College of Pharmacy, Belgaum, and on behalf of KLE Academy of Higher Education and Research, we heartily thank you, sir, for accepting our invitation. And in future, will be in touch for any sort of such scientific sessions.
Sure.
Thank you.
Thank you. Bye.
Sir, request you to, we are sending you the Google form for feedback.
Okay.
Fill it and submit it.
Okay. Thank you.
Thank you, sir.
Bye.